for being with us today. Welcome to everybody to the third seminar of HITRIplus. And before starting, we kindly ask you to turn off your camera and microphones in order to allow a better connection. So please, I will introduce Professor Haberer. Professor Haberer is the Scientific and Technical Director at HIT Center, and he has devoted his scientific career to the interdisciplinary field of ion beam tumor therapy. In particular, he introduced a crucial innovation in particle therapy, the raster-scan technique. In the initial phases of Professor Haberer's research, raster-scanned carbon beams were produced at the Germany's IV Ion Research Center, GSI, to treat with excellent clinical outcomes more than 400 patients, predominantly suffering from tumors at the base of the skull. Afterwards, the first hospital-based ion beam therapy center at the Heidelberg University Hospital, Heidelberg Ion Beam Therapy Center, was established following the specifications of Professor Haberer and the principles of the raster-scan method. Since 2009, roughly 6,000 patients have been treated at HIT's fixed beam lines using its world-unique gantry with proton and carbon pencil beams. HIT's research infrastructure allows for the constant development of the technology for ion beam treatments. In particular, it recently saw the introduction of novel treatment protocols based on helium beams. Today, the beam scanning technology is widely adopted in the field of particle therapy. So I leave the floor to Professor Haberer and enjoy the seminar. Thanks.
So I switch on my mic. Can you hear me?
Yes, we can, Thomas.
Okay. So I will take over the screen now. Let's see if this works. And try to start the talk. What do you see now? Is this the title slide?
No, you're not sharing the screen yet.
I'm not sharing the screen.
No.
Okay. So let's see, what do I have to do?
Just open your slides and then switch the green button.
Sorry, there is a dialogue for me. Okay. Okay. So let's see.
There we are. Yes.
Yes. Does it work now?
Perfect. Perfect. Thank you.
Okay. So I will start. Whenever you have a question, you should interrupt me or collect them for the discussion section afterwards. I'm prepared to all of these
thank you.
variances. So I try to give an overview on HIT, where we came from, what we are actually doing in terms of some clinical issues, not all of them, the ongoing research and development, and an outlook. Everything started with the discovery of the Bragg peak in 1904. So it became clear that helium ions, namely alpha particles, are more efficient in ionizing at the end of their range, which is 116, 17 years ago already. The next relevant step for us was the introduction of high energy accelerators in order to be able to produce beams that can reach deep-seated tissue inside patients. In fact, there was a Nobel Prize awarded to a palm-sized cyclotron, some inches, but a big one. You see it in the middle picture, and you can even see Bill Chu, one of the pioneers in the field. Let me point at Bill Chu. Maybe some of the audience may know him. That was really used to treat patients. And on the right-hand side, we see some pioneers on the synchrotron development, which was at the end of the Second World War. And at exactly that point in time, Robert Wilson proposed to use charged particles in radiotherapy. That took some time, but in 1954, the first patients were started, and shortly afterwards at Uppsala. But it took some decades to go for the first hospital-based facility, which was, and still is, Loma Linda's Proton Therapy Center. And some years later, 1993, in Chiba, Japan, the first patients were treated with carbon. In the modern era, while the Berkeley projects were stopped and we took over, and in 2009, the Heidelberg Ion Therapy Center came into operation, and shortly afterwards, other centers like the Italian National Center, CNAU, some centers in Japan, Yogo, Gunma, and then Med-Austron, close to Vienna, stepped in. So we have a pretty broad landscape now. But if you look back, that is how it started. You see the clamps. That was really experimental. I don't want to go into the details. But what was the basis of these treatments? It was passive dose delivery. All these projects were based on accelerators delivering a single monoenergetic energy that was, the range was spread using modulation systems. It could be a range modulator like that to smear out the Bragg peak position, and then scattering systems, multi-stage, different materials, very elaborate, to produce a homogeneous dose distribution, which basically was a cylindrical dose distribution, much larger than the target. So they needed patient-specific hardware to somehow at least tailor the distal edge of the target volume at the expense of the proximal part. You had a high dose here, an unwanted high dose, and to block away particles that should not reach the patient. That already produced very interesting clinical results, but it clearly was a compromise. There's an excellent report from the Berkeley area published in 1993, compiling all these technological aspects. But one key problem was that the dose delivery could not be tumor conform. And that's already Berkeley gave hints what can be done, namely scanning the beam, using Bragg peaks that can be positioned inside a volume defined by the clinician only. So if this is the tumor and you see organs at risk that may be very close to the tumor or behind the tumor, typically with passive systems, compensators are needed, and they produce these high dose regions in front of the tumor, proximal to the tumor. And if you go for beam scanning, these undesired doses can be taken away. And you can even modulate the dose inside the target using these beam scanning methods, which frequently are named intensity modulated. In fact, it is dose modulation, what you really do. Anyhow, that is just wording. So we stepped in. In fact, this was my PhD thesis at GSI. In 1991, we scanned the first beam laterally, which was a xenon beam. And you see, this is still lab work. And a year later, we combined lateral scanning, these crosses, with cycle-by-cycle energy stepping, which would be a cycle-by-cycle range stepping. So you can step the Bragg peak position inside tissue, cycle by cycle. And that could be demonstrated in 1992 at GSI. So we had the basic functionality available to try to deliver doses into three-dimensional structures, volumes. In this already pretty old animation, you see how this works. You select an energy from the synchrotron. You use a focused beam monitored in a series of patient monitors, position sensitive. And then you scan the beam over one slice of the target, one range inside the patient, and then slice by slice, you can fill the target with stopping ions, highly effective, and you can optimize this dose distribution. You need a radiological model to do that, and all these developments were in our hands at the mid of the 90s, and then we started a pilot project at GSI that finally treated 440 patients with carbon ions, predominantly skull-based tumors and salivary gland tumors. And you could see what this beam scanning method is able to do. You can produce very steep dose gradients. You can cover very difficult geometries with excellent sparing of organs at risk with this method. If you go for a full-blown center, which was part of the pilot project at GSI that treated patients from 1997 to 2008, and in 2003 groundwork started in Heidelberg, you finally have to combine a variety of ions. In our case, it's proton, helium, and carbon ions under clinical use, and for research purposes only, it is oxygen beams. You have to, or you may want to change the intensity fastly in order to minimize the irradiation time. Clearly, you want to have range stepping at about 1 millimeter resolution inside the patient for all these particles, and you may want to tune the pencil beam dimension in order to tailor the lateral dose fall off on the one hand side, or in order to minimize the treatment time if you use larger spots where applicable. So finally, you need about 30,000 settings per treatment room. And if you have a gantry, this multiplies by the amount of angles you want to use. In our case, at this point, it's 18 angles. So it's a tremendous amount of settings. So this can only be handled by accelerators of the modern era and very powerful accelerator control systems. So we managed to do all that, and we got the funding about 2000 and started digging the ground 2003 at the core of the Heidelberg University. This is our center. This is the central clinic here, where the conventional radiotherapy department running 8 photon accelerators for radiotherapy, treating about 4,000 patients a year. The radiology, the diagnostic department. This is Children's Hospital, Women's Hospital, internal medicine, surgery. We have the Heidelberg University here. We have some spin-offs here. So this is a pretty big campus. And we are very happy that we are at the core of this hospital, because it really allows treating inpatients under anesthesia, patients that have in parallel chemotherapy, and not being restricted by these given facts for the patients. So it's an ideal situation for such a center. From the facility point of view, that is how our system looks like. We have 3 ion sources running 24 hours in parallel. We have a carbon, a helium, and a proton source. The oxygen beam is experimentally used only and is extracted from the carbon source, driven by carbon dioxide gas. We have a 5-meter Linux structure that accelerates beam to 7 MeV per nucleon, and then injects us in our synchrotron, which has a diameter of 20 meters. And then these beams are extracted into 2 horizontally fixed beam lines. Patients are positioned robot-based, imaging systems are robot-based. And we have realized the worldwide first scanning carbon ion gantry. I'll come to that point later. And what is very relevant, on the right-hand side, we have a research environment with labs, radiobiology labs, technology labs, and an irradiation facility, which this year was equipped with an in-beam MR system in order to do some research with such an imaging device that hopefully later can be installed in one of our fixed beam rooms. This is our in-room CT. We have an MR system. We can shuttle patients already from our MR directly to the treatment couch. And we do research with an online MR, which is not yet patient ready. That may take some time. What you see here is the technology of this facility, the 3 sources, the LINAC, the beamlines, the synchrotron. And this is the gantry, where you see the structure, which is clearly a pretty big structure. It's an isocentric barrel type system. The beamline is seen here. The weight of the beamline is about 140 tons of magnets because the scanning system is located here upstream to the patient, and the already scanned beam is then guided with this 90-degree bending magnet down to the patient, which is this position, and this system can rotate 360 degrees around the patient at a speed of 3 degrees per second. And as we use edge focusing in this magnet, we have minimum divergence in the beam, which is very comfortable for the treatment planning and the QA procedures. But it is a very big device because everything is conventional technology. The system was designed in the years 2000 to 2004. Those days, superconducting magnets were not feasible for that kind of application, so we had to build it with conventional magnets, but it's heavily used by our clinicians, and we hoped to build 2 of them those days, but the funding finally was not available. So we realized one after a prototyping phase at GSI. So already at GSI, we had a horizontally fixed setup of the last part of this gantry beamline, and we scanned within the full range of the energies through this segment in order to understand the ion optical properties of this scanning system in combination with this large aperture bending magnet. And we already did raster scanning with helium in 2004 at GSI. In fact, this is exactly the elements that are used at our center today. The magnet was already usable, so we made this magnet rotational and installed it on our gantry. In October 2012, the first patient was treated. It was an astrocytoma. On the left-hand side, you see a tomotherapy plan. And you see a 3-field carbon ion plan for these scanned beams. And you see how excellent the dose distribution with carbons was those days. And hopefully this video starts here. You can see the device rotating. You have seen some person standing there to give you a scale. And just to give you an impression how this gantry rotates, that is the typical speed, but the nozzle was not installed when this video was taken. So our operation since the start of the treatment in 2009, it's pretty constant. Every year we have operation hours of 8,000, more than 1,000 shifts per year. The days of operation technically are 330. This is this line here. We have about up to 270 treatment days every year. We have treated in the order of 700 patients over all these years. That was our maximum patient throughput, 724, I guess, in 2015. Afterwards, we used a bit more time for the helium commissioning, but we are always close around this 700 patient margin, which is not a technical limit. This is just that we do not want to treat patients during the night, which could be done technically, but that is not what our clinical team would like to do. We have treated 7,100 patients since 2009. We treat 50 to 70 per day. The mixture proton carbon is about 50-50 these days. Close to 45% of our patients are treated at the gantry, which clearly demonstrate the clinical necessity of the additional entrance angles. Finally, if you produce them with a rotating beam or you rotate the patient, which is not feasible today, but may be available in the future, that's not relevant, but you need these additional entrance angles. That is a clear message. Our synchrotron has been used already for about 24 million cycles, and the gantry has already rotated about 300,000 times. So this system is heavily under use, and we already see some aging effects, but we are confident that this system can run another 20 years. 36 hours of patient treatments and 18,000 hours of research and development were performed at this system. We have a long-standing tradition in running clinical trials. These are the trials recently finished. Partially they are published, partially they are under analysis. These are the indications or the locations: sarcoma, central nerve system, head and neck, prostate, And this is a rectal carcinoma trial that was closed. And these are the open trials. Potentially the list is not complete. Still, we are treating chordomas, chondrosarcomas, which were the first indications we started with at GSI, central nerve system, head and neck, Pancoast tumors of the lung. They are not really moving. That's why we are treating them. We are not treating fastly moving tumors these days. And then we have pancreatic tumors, hepatocellular carcinomas, and prostate tumors. So it's a very large variety. I want to go through about 2 or 3 clinical examples now. This is a trial on salivary gland tumors, which was started at GSI. It is a boost concept where we treat on a photon bath of 54 gray the patients with a carbon boost of 18 gray, which is 6 days of treatment at 3 gray equivalent with carbon ions. We have significantly higher local control in the carbon group, and the overall survival after 15 years, we have 15 years of follow-up for these data, and this shows that it takes a decade or maybe 2 to finally collect all the data of these studies to draw conclusions. So it's not that you can change the clinical settings within the timescales of 2 or 3 years. You need to analyze and follow up patients for a long phase to finally draw conclusions. In this case, we are lucky that we could follow up some of these patients, the dominating part, for 15 years. And what we see is the overall survival is about 3 times higher for the carbon boost fraction of these patients on the one hand side. On the other hand side, we see some distant failures, not inside the treated volume, but distant. So the new protocols combine chemotherapy with particle therapy for these patients to even improve the outcome. This is a clinical example. Here you see this massive tumor prior to the treatment. This is the carbon boost plan. And this is the situation 6 weeks after the treatment. You see that already anatomy has dramatically changed to the good. That shows how powerful these carbon ions can be. And in the next stage of these studies, the ACCO trial with this, which is adenoid cystic carcinoma and carbon-12 only, ACCO, pretty crude name. We compare carbon only relative to a carbon boost concept and want to see if this additional high LAT part in the overall treatment protocol will improve the output. Another trial which is still active are the skull-based chordomas. This is one patient we in fact treated already at GSI. Here you see the tumor on the left-hand side at the base of the skull. And you see the follow-up over another 10 years. You see the structures are perfectly healed. And what we see for this fraction of patients, it's a rare bone tumor. Resection is frequently incomplete, and this tumor typically is radioresistant, so it's at high LAT, namely a carbon indication, or maybe in the future oxygen in our center. But this is not what we want to start soon. Nothing we want to start soon. And typically cranial nerve symptoms are recognized by the patients, and then they enter the hospitals and some imaging is done, and we can then start treating them. So 79 patients were treated after biopsy. The median follow-up was 91 months, and the local control was 88%, and in the younger fraction of these patients, it's already 98%. So you have to distinguish between these subgroups, but that is an excellent outcome. That's why we still treat these pretty rare tumors, but we have quite a lot of these patients in Germany, and they typically are directed to our center. A very motivating fraction of our patients are the pediatric patients and the young adults. Clearly, in the very young patients, We do everything we can to reduce the integral dose to non-target regions. We try to escalate the dose, and clearly we want to minimize late side effects. One impressive example you may have seen before is the medulloblastoma. On the right-hand side, you see photon treatment plans. And obviously, the intestinal organs receive pretty high doses, up to 20 gray, and the target dose here should be in the around 30 gray. And if you make use of the Bragg peak, namely the stopping characteristic of the particle beams, which can be tailored perfectly to the spinal cord limit, the dose to the organs at risk here is about 2 orders of magnitude lower, 1.5 orders of magnitude lower relative to photons. And this is very relevant. Coronary heart disease, for example, is a frequent finding here, and the additional relative risk for this late side effect is 7.4% per gray mean heart dose. So it makes very much sense to use particle beams in younger patients. It makes very much sense to use particle beams, at least I'm convinced of that, in left side breast cancer in order to spare the heart. This is our weekly schedule. What you see here is the patient treatment slots, 6 days per week. We have some QA in the morning, in the evening, not every evening. And then we have experiments running every night and during the weekends. And especially these slots here on the left-hand side, the night, are used for further optimizing our system, bringing in new functions, and to run research programs, internal and external. We are connected very well to the research community in the field, but we have our own research teams on the campus. So we are typically fully booked and have already used 18,000 hours to run these experimental programs. Recently, we have concentrated on helium. These are teaching pictures of radiochromic films positioned at the skin level, the upper row, and at 20 centimeters water equivalent path length. For carbon, you see that the lateral scattering of carbon is very, very mild. So every structure of this signum here of the Heidelberg University can be seen even at 20 centimeters depth in water. Helium is close to that, not perfectly, but what you see is a fully blurred picture for protons. So the precision of helium clearly is superior, and we have invested quite some time to commission helium beams to treat young adults and potentially meningiomas, for example, with helium beams in the future. The first part always is the physical beam characterization in order to have the pencil beam library perfectly described, or almost perfectly. Everything is then modeled. We use FluCA in order to produce the base data for the treatment planning system. We had to run through nuclear cross-section measurements, which was a cooperation with GSI, in order to improve the cross-sections for these Monte Carlo calculations. And this was published, I guess, a year ago. And in parallel, we typically study the relative biological efficiency of these beams. This is a comparison in various cell lines for protons, photons, helium, carbon, oxygen. What we found was an RBE of 1.7 for these helium ions. And this always is modeled with Monte Carlo means. And typically we run clinical protocols in small animals like mice and rat before we start a new modality like helium in patients. We did planning studies, helium versus protons. In this case, it was a meningioma patient, which was replanned with helium. And easily the target could be covered in a similar way, but we saw improved sparing up to 9 gray in regions close to the tumor. So we have less dose to the normal tissue, less dose at the brainstem. You see the dashed line is the proton DVH, whereas the full line is the helium DVH. Helium beats protons clearly, which is part of our motivation to start helium treatments. This is a tumor, a more practical case, because it's something that may be part of our first helium studies, a tumor at the chiasm, at the optical system. The brainstem here is irradiated relevantly higher than with the helium plan. And in fact, on July 20th this year, we have treated the first patient with helium with a raster-scanned beam, so with a beam of the modern era, after about 2,000 patients have been treated in Berkeley with passive systems. Everything we do is accompanied by a strong modeling group led by Andrea Mairani. And this team has developed very powerful tools to model whatever is needed, various biological targets. They run different radiobiological models, the local effect model from GSI, flavors of that, a modified MKM model, that is what the Japanese colleagues use. And using these powerful tools we have, all our studies are well prepared. This is another example where the biologists use this 96 well cell cultures to study the effects for various cell types. And this is a comparison of 4 different radiobiological models by this modeling group, and this really helps us improving our clinical protocols. Another flavor is the mixture of ions in, for example, in order to produce plan with a constant RBE, with a constant radiation quality, a clinical quality, which is not the case if you use carbon only, for example. You see clearly a variety of the RBE in field, which may not be the optimal condition. So in the future, we could, for example, mix helium and carbon beams in order to harmonize the RBE over the full target volume. Very interesting study they did recently, which was Stuart Mayen, part of this strong modeling group. It's a study on arc therapy with hadron beams. So what we do with our patients today is we use all these angles that are colored black here, which is about 18 angles that are used frequently. Whereas Shark wants to rotate the gantry while the beam is pointed into the tumor. And this arc therapy would allow for superior dose distributions. And here you see the angular fluence maps for proton, helium, carbon. For a clinical example, you can imagine this would be a chordoma at the base of the skull. And this is a cut through this dose distribution here. And you see the red line is helium, for example, which is potentially the highest quality in this dose distribution. The proton beam clearly gives higher doses to the normal tissue, may not be excellent. And due to the very sharp lateral dose fall of carbon is much more precise here. And what you see, if you use this, if you could use this modality, it's just a study, that you can select the LET value, so the radiobiological property of this beam, with the selection of the particle. For protons, it's clearly a very low LET value you will see, whereas in the planning target volume here for carbon, it's in the order of, let's say, 60, 70 keV per micrometer. For the protons, it's below 10. And for helium, it's close to maybe 15, 20 keV per micrometer. So the clinician would have the option to select the ideal LET, RBE distribution for those patients. We have compared it against volumetric arc therapy with photons, 6 MeV photons, and Shark clearly outperforms the VMAT plan here. You see the DVH coverage of the CTV is similar in both cases, photons and Shark, whereas the organs at risk dose is dramatically worse for VMAT plans in the chiasm and in the brain. The shark brain DVH is more or less exactly on the chiasm line. That's why it's not easy to see. And the dose distribution is superior too. So it's a highly attractive modality. It allows for robust planning. Normal and organ at risk tissue can be spared. Maximally, the LET distribution can be tailored to the oxygenation status of the target once it's known. So if the clinicians tell us where these hypoxic areas are, we could put high LET bragg peaks inside these parts of the volume. On the other hand side, you have to invest irradiation time, which may not be optimal. And we clearly have technical challenges ahead, because once you start rotating your beam around the patient, you want to be sure that if the beam is switched on, nothing moves. That is the way we run our system today. And if you want to move the gantry and a guide beam into the patient, you have interesting safety issues which are not solved. But on the long run, I'm convinced these systems will be available and maybe even part of the HITRIplus joint research activities. There is a gantry pillar in there. That could be an interesting part of that study, to find a safety concept to rotate the gantry while the beam is switched on. This is a graphic showing the beam usage. You should just concentrate on this lower row here. This is, the red one is when the first horizontally fixed beamline requests beam. The blue one is the gantry, and the green one is the second horizontally fixed beamline requesting beam. And these white spots here, this is when our synchrotron runs idle, which is something you don't want to have in your system, though there is room for optimization in the positioning, in the imaging, but in the irradiation and acceleration process itself. And you see this was one of our typical treatment days from 8 in the morning until 8 in the evening. System is fully loaded. And we brought in one of these development issues into the HITRIplus joint research pillar, which is the work package 10. We want to use the circulating beam for not only one slice through the tumor, but a series of them in order to bring down the treatment time. HIT, in fact, participates in 4 of these working packages. One is clinical networking. Then we have this transnational access work package, where about 10 patients can be treated at HIT, and external clinicians can be trained. And about 70 hours of beam time in our research room can be given to external researchers. Then we have this multi-energy extraction system that we want to develop, and our modeling group will participate in the radiobiological work package 12, headed by the Marburg colleagues. So what is the idea behind multi-energy extraction? Here you see in an ideal situation, the scan, the focused beam, the detectors monitoring the focused beam. These are the scanning magnets, the pole faces of these magnets. And here you see an artistic view of the slices of the target volume and the series of bragg peaks needed. What we do today at HIT is we inject beam into the synchrotron, we accelerate the beam, we extract the beam, and while we extract the beam, it is scanned over one slice. And then we deaccelerate the beam. We have to do some control issues in order to prepare the system for the next cycle. So it takes about 3 seconds, sometimes 5 seconds, sometimes only 2 seconds to come to the next extraction. So if we could use the already pre-accelerated beam, re-accelerate it for the next slice, so we could then extract various energies within one synchrotron cycle, that could save the irradiation time relevantly. So it could save about 40%. And we have already tested it for one change of the energy in the cycle. That is what we could do with our production system. The next steps have to be prepared in a research system, and a part of this analysis can be done within HITRIplus. But we could already show we extracted a beam for the first section of this spill, extracting 352 MeV per nucleon. We did a Bragg peak measurement, a highly precise Bragg peak measurement. Then the beam was re-accelerated from 352 to 358 MeV per nucleon. It was a carbon beam. And the range shifts by 6 millimeters. There's an M missing. So we have a perfect stability in these beams. So this is kind of a proof of a principle. And now the system has to be tailored to the real production needs. And that takes some time, but hopefully we make this progress. Another interesting issue, and clearly it is on the hot list of all the particle therapy centers, we need imaging inside the treatment room. We want to see what we treat. As a first step, we added a sliding CT, which can slide over our treatment nozzle here in order to have volumetric information before we treat the patient. That can be done on a daily basis or once per week, whatever is needed. And the relevant of margins that can then be boiled down once you have a daily image is clearly to be seen here. This orange, the peel, represents almost the same volume as the fruit itself. So reducing the margins is very valuable. Similar issues you can have with MRI, where you have real anatomic information in the imaging. If you look at this comparison, this is the initial planning CT, and this is a CT taken 5 weeks later. It's a lung tumor. The shrinkage during the therapy is relevant, so you have very negative consequences for the dose distributions because you have overshoot here. So it makes very much sense to have imaging inside the treatment room, and that is why we kicked off a project named Artemis, MR-guided ion beam therapy. The aims are we want to capture anatomical changes. We want to potentially track movement with MR. That can be done offline, and then we have to shuttle the patient, or in the long range online. And we want to provide optimal protection of the organs at risk. Artemis aims at a demonstration system, not a full-blown clinical system, but which could then be used to build a medical product on that. And the vision would be not turning the patient, but the beam. Even though we have a gantry, it makes sense to invest into this field. We have, for this purpose, in our research irradiation facility, where those who are interested in TNA potentially can use our research beam. We have added an online MRT from an Italian company, SAOTA, which had a pretty low field value in order to study the effects of the combination of beam scanning with charged particles in an MR field. As a first measurement series, we had the scanning system running and took pictures of this fruit without the scanning system, with the scanning system running a carbon beam, and you see the image quality is perfect. In the next step, we want to study how the dose distribution interferes with the MR field, and if we can correctly predict the corrections needed to return to an undeflected dose distribution once the MR is running while the beam is scanned into a target. What is the situation worldwide? This landscape is still a bit disappointing. As you can see, there's practically no facility in Africa. It is concentrating on Central Europe. In the US, we have protons only, and then we have these hotspots in Asia, especially in Japan. And you see even carbon therapy is evolving at a pretty good speed. Yeah, we will have about 12 to 15 carbon facilities. Finally, it may be 5 in Europe, 4 are already running. And the number of patients treated with protons is rising sharply. Carbon is a bit behind, but it's still frequently used. And what clearly would help is offering more compact, hopefully cheaper systems to mid-size hospitals. This is a proton system with a very compact proton accelerator. But there are already ideas out, and this is another work package within HITRIplus to build a very compact, potentially superconducting based synchrotron for carbon. And then you can potentially imagine tennis court size carbon facilities, single room facilities, and that would really be very interesting for the radiotherapy community in the world. If you compare this with the standard, let's say 2 or 3 gantry setups for full-blown facilities, they make only sense at very large hospitals, whereas this can be relevant for another fraction of radiotherapy users. If you look back on a very long range, this is what photons did in 1980, just 2 opposing fields. This is what particles can do 2020. And HITRIplus hopefully helps to bring in the next step in technology and the next step in radiation quality improvement. Ion beam therapy clearly is extremely precise, but it is challenged by costs. It is challenged by organ motion. The mitigation is not yet perfectly solved, but the huge potential is evident, and hopefully these developments are ongoing for a long period because we have so many patients that can benefit, and HITRIplus can help. Thank you. So I step back.
Okay, thanks, Professor Haberer, for the brilliant presentation. And before leaving the participants the space for questions, I kindly ask to everybody to turn on the camera in order to make the photo, please.
And Thomas, please leave your camera on even for questions and answers so we can see you, please.
Yeah, but I may not change during the next minutes.
Oh, well.
Okay, we are done. Thank you so much.
Thank you, Petya. Okay, let's please have your space for questions.
Sandro has a question. I can't hear you.
Yeah. Sandro, you're silent. You can ask questions by putting your hand up or by putting it in chat. So while we wait, Sandro, while we're waiting for you, maybe we can ask Eugenia to ask her question.
Yes, hello. Thank you very much for a very interesting presentation. My question is about downtime. During the days of operation, how many downtime due to malfunction or issues in hardware did you have? And what is the system which deemed to be the most fragile in the overall design?
We have neglectable downtimes over, what do we have now? We have 12 years of clinical operation. During 12 years of clinical operation, we have lost 2 days of treatment.
Wonderful.
2. What we have frequently, let's say we lose half an hour here and half an hour there. That may happen 2 or 3 times a week. We have 2 technicians taking care of the irradiation systems in the room and the imaging systems. They are sitting in our center, so if anything fails there, they just go downstairs and typically can help very soon. Yeah, so there is no identified system that frequently fails. the task is, once a system fails, it may be an error you see for the first time after 10 years, and then you need a team that has the expertise to understand this error. You have to have the spare parts around, and you have to have the expertise to repair it. That is what we could do, because part of my team developed the system.
Okay.
The accelerator system is in-house made. We built it on our own. We have coupled it to the irradiation system by Siemens and the planning system from Siemens, and lately a research planning platform. So we have all the experts on site, and typically we can resolve these problems soon. For example, we had to change the radio frequency structure of our injector, which is the first 1.5 meter of our linear injector. We had a spare part which was commissioned and stored, and within 6 days we came back into operation. Luckily enough, not all of these 6 days were planned treatment days. So if you have a trained team and all the spares around, you can help yourself pretty soon. Yeah. So our availability of the system clearly is higher.
It's very high.
If
yes.
you compare it to the conventional photon system in the next building. Now we have 8 photon systems, commercial systems for conventional radiotherapy. Our availability is higher than what the industrial partners here typically deliver. Yeah.
Thank you, Thomas. Sandro,
Thank
are
you
you
very
ready
much.
with you?
Let me see if now it works.
Ah, it works. Great. Thank you.
Okay. So I was saying that I always appreciate Thomas' talk. It was, as usual, very, very, very complete and wonderful. My question concerned the NMR system that he has shown toward the end of the talk. I would like to understand where stands the development work concerning the interference of NMR with the beam delivery system, with the raster scanning, and if they want to use it online during the treatment to make the tracking of the tumor.
Mm-hmm. So we have this small NMR system installed into our experimental irradiation facility. I showed the figure. This system is now running. The scanned beam is injected through a shielded window. And what we have tested so far is that the MR system, once the scanning system runs, gives us images that are comparable in quality with a undisturbed MR system. That works. We have measured, which is not a very difficult measurement, everyone can calculate that, how the pencil beams are deflected once the field is active in the MR system. This is mild deflection, and we are in a phase actually that we can model the deflection of the scanned beam inside the an experimental treatment planning platform in order to correct for the deflection produced by the MR. This development is underway. We have a prototype of the algorithm running, and I guess within the next 6 months we will run first irradiation trials to produce 3D dose distributions with scanned beams inside this small MR. So this is very basic work at this point, and another group is studying how a patient could be handled inside an MR that is part of the treatment room technology, which is a study. And I don't know the details here, but they have developed some of this positioning equipment. Yeah, let's see. So we have another, about 18 months, I guess, for the first funding period. And then we'll collect all this information, and hopefully we get a funding for another 3 years. And then we should be able to define how such a system finally should look like to be integratable into a carbon ion or multi-ion treatment center like HIT or CNAU or whatsoever. In a first stage, clearly, this should be located in a horizontally or vertically fixed beamline. Does not make very much sense to try to put it into a gantry beamline. Yeah.
Okay. Thanks, Thomas. So Emanuele next, and then Alberto. Emanuele, turn your audio on, please.
He's
Emanuele?
not listening, I guess.
Okay, so let's get Alberto De Giovanni to ask the next question, and then Emanuele can come in. Alberto?
Yeah, hello. Thanks for the very nice talk. I just had a question out of curiosity. Is there a reason behind the choice to go from the low energy to high energy in the multiple energy acceleration within the spill, rather than going from high to low? And if there is a reason, which one it is?
The very simple reason is we step upwards in energy all of the time. And we don't want to change this systematic, because multi-energy extraction mode should be an extension to the already existing controls mechanisms. And we have to clearly couple the accelerator controls to the therapy controls. And we do not want to touch this protocol at all, because then we would change the medical product installed in the treatment rooms doing the irradiation process. And this is a pretty high threshold. From the radiotherapy point of view, it does not make very much difference if you step upwards or downwards. Once we had a PET system in beam running in the pilot days at GSI, it made sense to start at the distal edge with the highest energy. No, let me think. No, it's the wrong way. We started at the proximal part, because then the Bragg peak always is located in an area where no activity is deposited before, which gives us more in-beam PET quality. But as we have terminated these PET activities, at least in HIT, that is no longer a reason.
Thank you.
I have Emanuele now. Please go ahead.
Okay, thank you. Thank you, Thomas, for a wonderful overview, as usual. What I wanted to ask is about the hadron arc system. So is the angular choice also part of the optimization, or
Yes.
it's just?
No, that is fully taken into the optimization to find these angular fluence maps. And the interesting part could be, if you put in oxygenation information in the volume, we could really tailor the LET distribution such that we have very high LETs in the hypo-oxygenated parts. That is, from my point of view, I'm not a radiobiologist as you are, at least you have much more understanding than I have. Should be very interesting to have the capability raising your LET up to or whatsoever 100 keV per micrometer in some volume elements there. That should really help.
Really flexible. Yeah, absolutely. And in this connection, so you plan to use this also for the oxygen beam, or is of course, I mean, this is very far to be included.
Exactly.
Yeah.
At this point, it's a study. We have safety issues if you would try to rotate the gantry and have the beam switched on, which annoys the researchers clearly. We analyze if we see a way to overcome that problem, but if we would, there is no limitation in what particle would be used with this mechanism. Okay. But so far we use protons and carbon regularly. Helium, we have just started the first patient, and we are now defining the first series of studies, which are for formal reasons named clinical investigations, because we have to demonstrate the feasibility and safety of the helium operation, and then we get a full approval for that. But at a certain point, we may add oxygen too.
Okay.
Especially for gliomas or glioblastomas, this may be an interesting option, yes.
Thanks,
And as far
Thomas.
as the very last...
No more. No, there are 3 more questions. Sorry. I'm being...
I have time.
No, but do you...
Can I?
Okay, go quick.
The switching times between the particles is comparable to the Japanese one or not?
You mean if you change from, let's say, proton to carbon?
Exactly.
That takes place synchrotron cycle by cycle.
Okay.
So you can have one extraction, let's say 5 seconds of beam, 2 seconds of preparing the beam, and the next 5 seconds could be another particle. Very fast.
Okay.
He's frozen now.
No. So, Dick, are you still there? Please go ahead, ask your question. Vikram?
Yeah, I just wanted to know, can you hear me?
Yes, we can.
I can hear you.
Yes.
Yeah. So what is the status of enrollment in the adenoid cystic carcinoma randomized trial? How many patients have enrolled already and how many more are you looking for?
Oh, I don't have the numbers. Let me have a look if there's something on the slide. This was this ACO trial. Okay. Pum. So Cosmic were 45 patients. ACO may be in the order of close to 100 again. If you want to have the exact number, would be nice to send me an email and I can find out.
Sorry, your question was already answered about oxygen. So the next question is Mimosa. Please go ahead.
Hi. Hi, everybody. Thomas, this was a nice talk. Yes, it was. I was always puzzled about the chemical changes that are induced by certain particles.
We can't hear you, Mimosa.
I cannot understand the question.
Like hydrogen.
Mimosa, turn the video off and then ask,
okay.
Please.
Okay, so using different ions in a certain tissue, it's not just breaking the DNA chain, it is also inducing some chemical reaction because you have introduction of certain elements that should do something there in the environment. Is there any study that shows shows what are the chemical changes in the environment, for example, in a bone tissue? What happens if you have either protons, which are reactive, helium ions, which are inert, and carbon, and oxygen, which is oxidizing.
So
is there any chemical?
You should have a look at the absolute number of particles we bring into the patient, which in carbon, those distributions can be up to 10 to the 9th. And a mole is, I don't know how many orders of magnitude more particles. So there practically no relevant amount of molecules could be built. That is why I do not have any information on a single study taking care of chemistry with these small amounts of ions. Should be neglectable.
Okay, thank you.
So if there are no other questions, may I ask a question, the next one? Anybody else? Okay, so Thomas, so I've seen that you've treated your first patient with helium in July. What are the plans for that in the near-term future?
We are now preparing the approval formal documents for this next phase, which is called clinical investigation. So we have to define the number of indications and patients that will be treated with helium. Then we will analyze that, and then we can use helium in a broad picture of clinical trials. That should happen So the start of this clinical investigation should happen within the next, I would say, 6-8 weeks.
Okay, thank you. So if there are no further questions, let me thank you, Thomas, for an excellent presentation. And
You're
Sylvia
welcome.
will close the seminar. And I just remind everybody, we started off with the clip on the transnational access. If you're interested in trying to use the beam time, please have a look at the video and also the link, because this is a possibility to join the HITRIplus collaboration, both for research and for clinical aspects as well. So Sylvia, please.
Yes, we showed the video before, and we are very glad for the results, for the result of this video. And now we thanks especially Professor Haberer for his presentation and for the data he shared with us, and for all the attendance of this seminar. So we see you at the next seminar.
There is one question from Giuseppe. Do we have time? Thomas, do you mind?
I have time, yes.
So Giuseppe, please. Sorry, Sylvia.
Hello, Thomas.
Hello.
Just maybe I was distracted. I didn't get from your seminar. Could your gantry be used also for helium?
Oh, yes, it's already commissioned for helium.
Okay.
Yeah.
So all the 4 ion species in principle can go through the...
No.
No.
Proton, helium, and carbon can be used in the clinical rooms, the 2 horizontally fixed beamlines and the gantry, whereas oxygen is only commissioned for the research facility, the research room.
Okay. Thank you.
Sylvia, sorry. Go ahead again.
I think that we can close the seminar and leave the attendance to their families.
Well,
Okay.
Thomas, there were lots of questions because the talk is really interesting, and of course the subject is really developing. So thank you, everybody. We look forward to seeing you all at the next seminar. Thank you.
Yeah.
Thanks, Sylvia.
Bye.
Thank you.
Bye-bye.
Everybody. Bye.
Bye.
Thank you.
Thanks, Thomas.
Bye.
Bye.
Bye. Thank you.
Bye-bye to everybody.